Welcome to the final presentations of the Summer Undergraduate Research Symposium or Program. I think as I was thinking back about this, COVID-19's interrupted a heck of a lot of things. I'm really pleased that this is not one of them that was impacted. Being able to see students and faculty working together on scholarly, real research projects, it's just one of the very, numerous examples of things that make this place so special. We do have 5 presentations to get through today. So with that, we're going to get started. The first presentation, we have faculty member Elizabeth Coody, and we have students Alexandra Inskeep and Keonté Robertson. Their project was entitled The Comics and Sacred Text Database Project. So let's welcome them.
All right, so hi, my name is Alex Inskeep. I am going to be a sophomore this year at Morningside, and I am a double major in religious studies and counseling psychology.
Hello, my name is Keonté Robertson. I'm a computer science major, and I'm a junior here at Morningside, and this is our Comics and Sacred Text database.
All right, so first off, we're going to define what comics and sacred text is for you, just in case you didn't know. So for us, comics have been around for ages. I mean, as soon as we could use pictures to tell stories, that's how long they've been around. But today we see them not just as superheroes and villains and stuff. There's multiple genres that have been published, and you can access them virtually and in your own hands. But sacred texts, on the other hand, are religious stories that mean something to a religious group of people, and that are in words. So text as in books, paper, electronic devices now, all that kind of stuff. So let me see if we can click on the next screen real quick. All right, so you may ask, why did we create this database? So, we wanted to document instances between comics and sacred text and put them into words, because, we want to aid scholars who do this kind of work, who whether it be they study religion or comics in general. And we want to educate readers about this because we feel it is an important topic to talk about. It provides a place for people who are interested in this topic to reference to and to be able to contribute to as well. So one of the reasons we created this database is because it's not documented very easily, and one of those reasons why is because comics are art, and computers can't read art very well, so humans have to do this work specifically, and it takes a difficult time to track this stuff because of humans doing it and taking the time to publish them and getting them through to other people. So we created this database for people to contribute to and be able to do this kind of stuff because there's not many of those around. So during our time in this project, I was not a comic book fan. I barely touched one comic book before this project. So, coming in brand new, I had to understand the comics I was reading and how to make those connections between the sacred text and the comics. So I had to research a bunch of databases, including like a comic database called the Grand Comics Database. And I had to research a bunch of sacred text database too, because as a freshman, I only took maybe 2 religion classes so far. So, I was still pretty new at this. And I needed some resources to help me out. And so once we got those resources, and Keonté and I did a lot of collaborating with Dr. Coody on this, we needed to create an outline for what we wanted on our database. And so we created an outline for the entry form of what we were going to include and how we were going to include this. So for instance, like the relation between the comics was one of the things we talked about over and over again, because we have so many instances that can happen, like a sacred text, a plot from a sacred text could appear in a comic, a character could appear in a comic, even the images in the comic themselves can reflect a sacred text. So there were instances that we couldn't include and that were just kind of iffy, like we had to figure out what and when we were going to put it in there. And also during this time, we were thinking about the database itself, and we wanted something to be able to identify our database. And so we thought about, we need a logo, so what are some ideas we want with that logo? And with that came the domain name as well.
Also in month one, we started constructing on the database and using MySQL. The reason why we use MySQL is because it's one of the most popular databases. It's a relational database, which makes it easier for query, which is what we're doing here. We're querying to the database and displaying it to the user.
So some of the resources we use during our time here with this project is this is one of them. So this is Comixology. It is a company through Amazon that gives you people access to hundreds of thousands of digital comics. So Keonté and I both got unlimited subscriptions to this with our funding, and we got to access these comics 24/7, either on our computer, our tablets, or our phone. So this was, as the reader of the group who was doing a lot of writing and stuff, this was super helpful for me so I could access these whenever I needed and whenever I wanted to. And so, I mean, you may think, how do we stay in communication? Because all the rest of the groups were in person almost all the time, and we were very virtual. I mean, we did Google Meet, which is just like Zoom, but through Google. And then we also use an app called WhatsApp, which is like a text messaging app where you could do quick texting to each contributor. Another resource we used was our Notion page. So this was a website that all contributors can access and edit. So we listed things like our weekly goals on this. We put resources that in the beginning of the month were very helpful for us. I'm trying to figure out how to read and do all this kind of stuff. Other databases like comics.org and other links to databases for ideas for our database.
So this is a Notion task list page. The best way I can describe this is like the road to success page. This is where we had stuff we haven't started, next up, in progress, and completed. We would assign jobs to an individual on the team. And this is where if you didn't do your work, it would stack up and you would be overblown with all the amount of work you would do. So yeah.
During our time at the end of month one, we kind of recollected on what we did, and we came to a pausing point on our outline and what we wanted on our database where we could tell people about this. So we did a lot of outreach to our friends, family, and other scholars and community as well about this database. I mean, I was telling my family 24/7 what I was doing. So, I mean, they were telling their friends and so on and so forth. But we did a lot of emailing, social media, and all that kind of stuff. And that got people to know about our database. And during that time, we also invited the graphic design program at Morningside, and we emailed all of them and we're like, hey, we need ideas for logo design. Who's willing to help us? And one of our very own at Morningside, Lex Worth, designed our logo, which you can see down at the bottom. And we fell in love with it automatically. Like, we thought it described our database perfectly. So we are very thankful for that. Another awesome thing that happened during this month is that we had a generous data donation from one of Dr. Coody's scholarly friends, Andrew Toos. He's had been documenting instances between the Bible and comics for years, and he sent us a bunch of documents with those instances on them. And all I had to do was go in and edit them, figure out what else I needed to find about the comic, and just send them into our database. I mean, it was so gracious. I mean, it made my job so much easier because I was reading full comics and then going and back and finding those instances and then going. So this was an awesome thing that he did, and we're so grateful for that. We also had a bunch of people emailing and reaching out to us saying how excited they are about our database, and even giving us ideas and like what we should put in our database and what should be next. So that was an awesome time for me, especially.
During month two, a lot of the time was spent troubleshooting and doing major website construction. When it comes to making a website from scratch, there's a lot of trial, plug and play, and you're kind of seeing what looks good, what doesn't. And there was a lot of troubleshooting with the database of how we were going to display it to the user, how it came out. So, next. This is the welcome page. This is what the user is greeted to. The whole website is made with PHP. We use PHP because it's the most popular, and it's open source, and it's based around object-oriented programming, which is very secure when it comes to like user security and usability. The welcome page has like a short about section, but we go next page. We have our big about section. This is where we try to convince you to for you to give us information, why you should search our database, why you should contribute to our database, and explaining that this is only phase one of the database. So if we convince you to give us your information for us to sell, you would come here and you would sign up. And once you sign up, you will be able to log in. And once you log in, you'll be able to make submissions of your own. So when you go to the this submission form.
So this submission form is basically the outline that we created in the beginning. So this has been the the structure of our database on a reading point of view. So it has all the essential information in red that we need. For instance, like the series title, the publisher, the series year. I mean, those are all super important to know about the comic. But the most important part we need about the submission page is the actual entry itself. So we need the name of the sacred text, the relation between the comic and the sacred text, like did that character show up, did that plot show up, and the entry itself. So what happened in this comic that the sacred text appeared in. So this is the biggest part of our database, and this is what we try to make the most important.
So once you make a submission, it'll be uploaded to the database, and it'll be there for users to find. Now users can search for it, or they can just go to our library, which we'll show in a sec. Once you click the entry, you can scroll and look at all the information and things like the name of the sacred text, the relation to the sacred text, a summary of the comic, and the entry itself, and external links that you use to do your research. This is the short little library where you can scroll and look through the catalog that we have.
So we have mentioned that this is only phase one. So you may ask, like, what is phase two? What are we thinking about? And really, phase two is we want more. We want more entries. We want more people to enter these things and for us to edit them and be able to put them into our database. We have a lot of certain types right now. So like the stuff that Andrew too sent us was a lot of the Bible, a lot of DC comics. So we want a variety of more stuff. So we want more independent publishers. We want a lot of other sacred texts than just the Bible, because there's more out there. We want everything basically. But during phase two, we also want to publicize this a lot more. So one of the things we didn't get to do this summer is we weren't able to go to Comic-Con, which while at Comic-Con, we were going to have a lot of fun. But one of the biggest things that we were going to do is we were going to publicize to hundreds of people about this and be able to share with people so they can know about it. And then maybe later, once we get this all finished up and in a spot where we can have people submit, they can go in and submit themselves. So phase two is really super important to us because we want to be able to make this a working and reliable source. So what did we learn during this time? I mean, personally, I'll share my experience first. I learned a lot about first theology. Like, this stuff can get super difficult. Like, at first I was getting frustrated because I don't know everything. So I had to research a lot. I had to go in and find out an instance and relate it to a sacred text. And I mean, based on how I was at the beginning, how I am now, like, I can point them out like this. I mean, I still need help, but it's my skills have broadened, which is super nice. And I also learned a lot about working virtually. I mean, we're in completely different states. I mean, I'm still in Iowa, but Keonté's not. So that was we had to meet virtually almost every single day, and working as a team, and being at home with all the distractions that those can have. I mean, it made some time management for me as well.
For me, this learning experience provided like a commission type work experience where someone has an idea and they come to me with it and I have to bring it to life. It provided some time to me with PHP and troubleshooting it in databases. And like Alex said, it did provide time for like us learning how to work from a distance and having people work from different parts of the country to make a project come together.
So we thank you so much for letting us do this project this year and taking your time today to even listen about this. So, we put so much work into this, and we hope it is inspiring to you. Thank you.
Thank you.
Okay. Our second project was by Dr. John Helms and Margaret Best. Its title, I'll give you the long title just because I had to practice it. Photochemical dissolution of phosphorus from soil runoff and resuspended freshwater sediments, how the unlocking of recalcitrant nutrients may lead to algal bloom conditions.
And that's why you should always revise your working title before you submit the proposal.
So I'm Margaret Bass. I'm a senior this year. And yeah, we did photochemical release of phosphorus, which is the easier way to say it. Our hypothesis was that suspended soils from runoff release dissolved phosphorus upon UV radiation. We did expect some dissolved phosphorus to release in the dark because it can be released by bacteria, by reuptaking and then letting it back out. But we expected a lot more to be released from the UV radiation. That's the whole point. In natural waters, dissolved phosphorus is extremely short-lived in the upper column because of the reuptake of the phytoplankton. So we're using UV-only light source so that photosynthesis would be minimized. So it would only be the photochemistry observed. So why should we even care? Does it matter if there's phosphorus in the water? Yeah, it does. That's why we researched it. Otherwise, we don't. In the ocean, surface biomass of carbon, nitrogen, and phosphorus are deeply dissolved nutrients are in the same ratio of 106 to 16 to 1. But when there's too much dissolved nitrogen or dissolved phosphorus, algal blooms can occur. And the photochemical release of the phosphorus can trigger algal blooms, which can be toxic. The low oxygen caused by the decomposition of the algal blooms can cause the fish and all the other natural life in the waters to just die. It's a huge issue. The dead zones form when excess nutrients like nitrogen and phosphorus enter the waters and help fertilize all of the algal blooms. The nutrient sources include fertilizers, wastewater, and the burning of fossil fuels. These algal blooms are often toxic in and of themselves, but the bacteria that break them down is what sucks all of the oxygen out of the water that can cause everything to die. This is the watershed of the Mississippi River, and that's the dead zone where there's not enough oxygen in the water. This is Lake Erie, which has issues with the harmful algal blooms every summer and can even impact the safety of the drinking water. So what we did was we first had to find the limit of quantification and the limit of detection for the instrument we were using. It's never really been used to detect phosphorus before, so we had to make sure it would work, which thankfully it did. We found that we can just barely see the concentrations of dissolved phosphorus at environmentally relevant levels. For the instrument we used, the samples are turned into a fine mist, which are put into the MPAES, and they get passed through plasma, and the electrons get excited and then go back down, and the wavelength emitted is read by the detector. water, which can tell you the concentrations and what's in there. So in order to do our sampling, we use the Van Dorn sampler to get water, which is this thing. And that's me just testing it out at the fountain. That's the easiest place to do it. And we collected samples from the Missouri River from multiple points. We went to Cheap, Big Bear Bridge, Standing Bear Bridge. And then we also went to the boat ramp in Sioux City. And then for soil samples, we went to First Bride's Grave, Dorothy Peacock, an alfalfa field near Rock Branch, and a cornfield near Mobile. Our results show that there's a consistent difference between our dark controls and our light controls. It shows that there's a lot more release when they're exposed to UV radiation. The photo release was almost always observed, but there were 3 instances where there was UV-induced loss of dissolved phosphorus. We also looked to see if there was a difference between the river water and the deionized water for the soil samples, and if there is a difference in how much was released. There wasn't. And then we also compared autoclave samples that were sterilized to non-sterilized samples, and there also wasn't a statistically significant difference between those different things. And that's it. Do you have any questions?
Who would use this information?
People working with farmers to try to keep any agricultural runoff from going into the water, and people who want to help keep the waters from having all those toxic algal blooms.
Scientists.
Are the fertilizers the main source
Um,
of phosphorus from the farmlands?
Possibly. There are other sources of it, but there is a lot more phosphorus in agriculture soil from the fertilizers than most other places it would come from.
So if there is a photochemical release of phosphorus, does that mean that we could speed it up by tilling or, well, shining UV radiation into the soil? Would that be
Possibly.
feasible?
But, yeah, but it is in a lot of other soils that would get runoff, so there's not always a way to make sure that it just dissolves out before it enters the water, because it would still just get in the waterway eventually anyway.
And the main way that you keep the soil particles out of the waterways is to avoid tilling it.
Exactly.
So,
I may have missed that. I was just wondering, what was the wavelength range of UV? Is this for all UV or certain portions?
it's very narrow bands. So the one we used for phosphorus was like 243.18 or something like that.
Any other questions for Margaret? Let's thank our speaker.
Hello everyone, my name is Dave. I'm sorry.
Our third project was a project with Dr. Stacey Alex and Damian Herrera, titled Latinx Stories of Siouxland and Oral History Archive.
Hello everyone, my name is Damian and I'm going to be a sophomore this upcoming year at Morningside. And our research project, Latino Stories of Siouxland and Oral History Archive, investigates the diverse experiences of the Latino population in Siouxland. Dr. Stacey Alex and I decided to work on this project because we would like to show how the Latino community in Siouxland has shaped the culture and lifestyle of our region, as well as the successes and hardships they continue to face. We have created a publicly available web archive of 8 life history interviews, which will also be archived by the Sioux City Public Museum, thanks to their Curator of Education, Teresa Weaver-Basie, and Director, Steve Hansen. Every group that makes up the Siouxland community is unique, and we would like to recognize their contributions that have been left out of many official histories about our area. I think that it is a wonderful project to partake in because I am Latino, and it is rewarding to demonstrate how hardworking and inspiring my community is and what we can accomplish. We are grateful for our participants' willingness to share their fascinating stories of adversity, success, and growth. This project was designed to showcase the diverse experiences of the Latino community in Siouxland. Our community makes up a huge portion of the people living in the area, and it should not go unnoticed. There are so many Latinos in Siouxland who have made a difference in the community. Their hard work has influenced the way our community functions and shaped our identity too. However, we do not want to limit Latino individuals to their role as workers. Focusing only on the economic contributions of Latino communities runs the risk of reinforcing the belief that we deserve unjust working conditions or that immigrant suffering is natural. Many of our interviewees talked about how hardworking we are to confront common stereotypes that criminalize or portray us as lazy. However, we do not want to portray the complexity, we also want to portray the complexity of our community. We are not just workers, but also students, parents, entrepreneurs, and community leaders. We have collaborated to create this public archive that will be stored and shared with not only people from our community, but for other people who are not familiar with Siouxland's history. We hope it will be used by people who are interested in either Latino culture, the Spanish language, etc., who would hopefully benefit from the information gathered from this research study. We provide a brief history of Latino presence in Siouxland that discusses interest in overcoming the lack of Latino representation in local official histories. So I'm just going to go ahead and show you our history page. Let's see, okay, and then here's some pictures, and then if you scroll down, it will give you just a brief history about Siouxland and the Latino community here. We also outlined some of the historical reasons that Latin American immigrants and Latino families from other parts of the U.S. have moved to the Midwest. Although Latino communities in the Midwest continue to face discrimination, there is also relatively less hostility in terms of state and federal policies, a lower cost of living, and job opportunities in a variety of industries. We also provide a list of resources so that our visitors to our website can find other Latino Midwest oral history archives. Now I will direct you to our resources page, and it gives you just a couple of links to go through just to get more information about everything. This information is provided both in Spanish and English. However, the interviews themselves are only available in the languages that the interviewee chose. This means that at this point, 7 are in Spanish and 1 is in English. We feel this is appropriate and celebrates bilingualism, but we may provide further translations in the future. Now, I will briefly go over the research procedure we have established. We've provided each participant with 2 consent forms to have permission to use the interviews for our project. The first form gives permission to record and transcribe the interview and make the interview available for public use through our website. The second form is to give the Sioux City Public Museum permission to archive the interviews and transcriptions so that anyone can view them if they wish to in the future. We made sure interviewees understood that they had the opportunity to opt out of the research study at any time and were not obligated to answer any questions they did not feel comfortable responding to. Our goal during the interviews was to make participants feel as comfortable as possible, as though we were having a regular conversation. While we developed a set list of 30 interview questions, we personalized the questions to focus on things we already knew about their lives, such as their professions or their studies. We also added follow-up questions in the moment to elicit more information and show that their stories are important to us. We reached out to various community organizations to invite the Latino public to participate in our project. These included the school district, a local church, restaurant, and Mary J. Traylor Community House. We also heavily relied on word of mouth to publicize the project and get recommendations for further interviewees. For example, one interviewee, Perla Alarcón Flori, recommended 2 other interviewees because she is a school board member and is well connected with our community. We were hoping to conduct the interviews live in person, but the COVID-19 pandemic meant we needed to interview through Zoom. Some people do not have access to the internet, so this limited the amount of people we could interview, particularly older generations who may not feel comfortable with technology. This will be an ongoing project, and Dr. Alex will add more interviews in the future, particularly to include older generations. We strive to make our pool of interviewees as diverse as possible. It is important to capture the variety of cultures that make up the Latino culture, or community. The Latino community has many cultures that are rooted in the already diverse cultures of Latin American countries, but also differentiate themselves from other cultures. For example, Rashmi, a graduate from Morningside College who we interviewed, explains how she was raised by her father from India and her Peruvian mother in a bicultural household, and it allowed her to gain multiple worldview perspectives. We also tried to have more interviewees of different age groups. Older individuals tend to have more historical background information and have lived through different time periods. They give us a perception of what life was like many years ago. On the other hand, yet younger generations offer a unique perspective on contemporary issues, particularly on entering and adjusting to the job market during the COVID-19 pandemic. While using Zoom wasn't ideal, we feel that it was important to continue with this project this summer anyway, particularly because it allowed us to gain an insight into how the Latino population has been disproportionately affected by COVID-19. Several interviewees shared their physical and emotional burden, losing employment or having family members contract the virus. In addition to the interviews being archived by the Sioux City Public Museum, Dr. Alex has also decided to make this website showcasing the work that we have done over the past few weeks. It includes all of the interview videos we have done. It will also have the transcriptions available to read and follow along to. The website will also show some biographical information about the participants so that anyone on the website will be able to know a little bit more about the interviewees. And then I'll go ahead and show you our people page. It takes a while. And then you can scroll down and then select any of the interviewees you've ever interviewed, and just gives you some background information as well. And then there's also an About Us page for Dr. Stacy, Alex, and me, so that people can see who we are and what our motivation for this project was.
Wait, Damien, scroll down. We gotta see how cute you are.
Yep, we'll go ahead and
We
show
gotta
you
see
some
how
of the
cute you are.
people.
There's
That's
Damien.
me. Some information.
We all know Diego, right?
Diego. Those are just some people that we've interviewed.
Thank you.
Yep. And then I'll go ahead and show you our About Us page as well. Here's us. And some information as well. And then pictures of the participants are also available on the website. If anyone wishes to participate in the future, there is a contact page for them to fill out so that Dr. Stacy, Alex can reach out and do more interviews. And the contact page is on our home screen.
You don't have to show my huge face if you don't want to.
I will. So if anyone's interested, they can just fill out the screen right here, and then Dr. Alex will be in contact with them. And then this project has greatly impacted my way of gathering information and asking questions. It has changed the way that how I perceive people. Everyone is different, and people have gone through many experiences that others have not necessarily have gone through. It is interesting to see how people have become who they are because of their experiences. It is important, in my opinion, to understand where they are coming from and why they think a certain way. To understand everything that has happened to someone is to know who that person really is. This research project has also greatly aided in my understanding of the Spanish language and Latino culture. Dr. Alex does an excellent job of helping not only revising my work, but correcting spelling and grammar, as well as organizing and conducting interviews to achieve our overall goal for this project. I've been able to improve my professional writing abilities and to use it to send emails in Spanish to contact potential participants about the study. Although I grew up speaking Spanish at home, I have never felt comfortable speaking Spanish, even at work, due to fear of not being understood or not being able to understand what the other person is trying to say. I have built confidence in speaking Spanish by asking and responding to questions. This study has given me the advantage of eliminating that barrier and by asking advanced questions that challenges me to think and respond using a professional register. The Spanish language is so diverse in terms of variations, accents, and rate of speech, and this project has given me the opportunity to see how diverse the Latino community is here in Siouxland. I've met people whose backgrounds range from Mexico to Puerto Rico and even to Peru. It is exciting to hear how different each person speaks because it gives you an idea of where they can be from. I believe that this project will help me in my future career of being a Spanish teacher for many reasons. I believe I will stay in Siouxland to teach Spanish for a couple of years, and it is important for children to understand the importance of the Latino community. This project can aid me in helping other Hispanic Latino students show the impact they can make anywhere, and specifically here in Siouxland. For example, one interviewee, Hugo Rodriguez, said that Latino youth don't have to leave Siouxland to make a difference, but there are a lot of opportunities here for growth. The project will also be beneficial for me and my students because it showcases how diversity can inspire new ideas and new connections with new people. This work inspires me to create assignments in which my own students can embrace the diversity of their communities. I want to help them establish their own identities, value differences, and celebrate who they are. I can also show them that everyone experiences adversity and they should not feel as though they are alone. I want to be a supportive teacher and have my students succeed academically and socially. I want them to feel as though that they can confide in me. I gained a lot of valuable skills from participating in this research study. I've gotten the opportunity to learn best practices for conducting interviews. I've also been able to meet different people from all over and hear what they had to say. I've also even been interviewed about the project by a local Spanish-speaking newspaper. This experience would not have been possible without the help of Dr. Stacey Alex. She has been great as far as helping me with transcriptions, preparing interview questions, and getting more context for our project. It sounds cheesy, but professors like Dr. Alex are one of the reasons why Morningside is the best side, and I'm so grateful to have worked alongside such a determined and caring person. Thank you.
Any questions for Damien?
I do. What did you use to build your website?
Oh, we used Wix. It was like a professional website site.
Builder?
Yeah, builder. Yeah.
And then tell me one of the most interesting stories you've heard that gave you a different perspective that you hadn't thought about.
Probably when we interviewed Rashmi. Like I said, she's a recent graduate from Morningside. She just explains about how she was raised in a bicultural household, and so since her parents are from, or her father's from India and her mom is from Peru, they had clashing cultures that were different from each other. So they had and she also how even inside a country they have different cultures. I mean, they have the indigenous people, they have other immigrants coming in from the country, such as from Japan, and so I mean it gets a worldwide perspective of the country and everything. So.
Thank our speaker again. Our next project features Dr. Annie Moore and Andy Hayes, and it's titled Comparison of Upper Respiratory Microbiomes of Pasture-Raised and Formerly Caged Chickens.
Okay. Hi, my name is Andy Hayes, and I'm a senior here at Morningside, and I'm majoring in biology and chemistry, or I'm a pre-veterinary student. And I did my project with Dr. Andy Moore back there, and it was titled, as he said, Comparison of the Upper Respiratory Microbiomes of the Pasture-Raised and the Formerly Caged Chickens, which was exactly what the purpose of this project was to compare the respiratory microbiomes between these two types of chicken. So why does it matter? So if you think back to the beginning of the pandemic, I think you can remember how you go to the grocery store and you couldn't find any eggs. So this was a big deal, especially in Iowa, because as it says up here, Iowa is the nation's leader in egg production, with 16.5 billion eggs produced annually. That's a lot of eggs. And so currently, all the egg layers in the U.S. are raised, or 90% of all the egg layers in the US are raised in conventional facilities, although like recently in the last years, and especially during the pandemic, there's been a large increase in people buying backyard chickens. So it's really important to keep these chickens healthy, because if they get sickness and spread it to their population, this leads to chicken death, and then again we might see a drop in the egg production and then no eggs at the stores again. So how do you keep your chicken healthy? Oh, never mind. Sorry. So you want to keep your chickens healthy, but obviously you can't prevent disease 100% of the time, because there are a lot of chicken pathogens. So here this shows a few of the more common avian respiratory pathogens and diseases that chickens can get. And these diseases not only can kill the chicken, but they also do have a large effect on the chicken's egg production. So it's a lot cheaper to make sure your chickens are healthy compared to treating them when they were sick. So now, how do you keep your chickens healthy? Okay, so the first line of defense for the chickens is their natural microbiome, and it is shown in many studies that the more diverse your microbiome is, you have more protection from infections and diseases. So here's a few pictures of some studies, and here on the left, this shows that those who had a more diverse microbiome here, they had milder cases of cystic fibrosis compared to the ones with the severe cases. They had less diversity. Up here in the middle, this side is the people with schizophrenia compared to the controls, and as you can see again, the people with schizophrenia have a less diverse microbiome. And then the pie charts here, this shows again that right here people that had less diversity were more likely to have disease. In this case, it was diabetes. So what influences the microbiome? Well, the environment that the chickens are raised in plays a key role in this. And there are 4 different ways that chickens can be raised. So here on the left, this is how chickens are commercially raised, and over here is the small producers are individually raised. Okay, so the top left, these are the cage chickens. So they have very little interaction. They're kept in their cages at all times. The only interaction they have is with whatever's in their cage and maybe their neighbor chickens. And then the bottom left here, the floor raised, so they have a little bit more interaction. They can roam around inside this building, but then again, they're still kept in this building, and they only have interactions with the other chicken in there and whatever else is in the building. The top right, these are the free-range chickens. So again, they have even more exposure to the environment and interactions. They're usually kept in like a small cage, but it's outdoors, so they have interaction with obviously the other chickens, and they can interact with the dirt and mulch, like you can see in here. So they have a little bit more interaction with more of like the environment. And then the pasture-raised chickens, they get the most interaction with the environment because usually they're allowed to roam on like an acreage, so they have interaction with not only the other chickens, but the grass, the soil, any trees or plants that are on the pasture, any insects that are in the grass. They get a lot of interaction. So knowing this, we came up with our hypotheses. Our first hypothesis was that the pasture-raised chickens would have a significantly more diverse upper respiratory microbiome compared to the cage chickens. Our second hypothesis was that the cage chickens would have microbiomes that were more similar to one another. And then thirdly, that the cage chickens would have a higher resistance to antibiotics that are used in commercial poultry facility settings. So how did we do our study? So first we started with getting the chickens, obviously. So we used pasture-raised chickens and then formerly cage chickens. The formerly cage chickens, they were born and raised in a commercial facility, but then they were rehomed to a more free-range setting, and we got them about 4 weeks after they were transferred to a free-range environment. So once we had our chickens, then it was time to sample them. So this picture in the middle, this is what the inside of a chicken's mouth looks like. I had no idea until the study. It was like super cool. It was crazy. And then, so what we did was we took sterile swabs and we stuffed these swabs inside this slit right here, which is called the choanal cleft. So we swabbed the inside of there to, which got the swabs into the nasal cavity then we took these swabs and then some of them we took to test for antibiotic resistance. Then the other ones, we took these samples and then we extracted the DNA from these swabs and then amplified the 16S rRNA gene so that we could use that to run it through our sequencer. And the sequencer we used was called the MinION sequencer. And then here's a couple of the different things we use for our data analysis. So this is the MinION sequencer, and no, it is not Minion, even though they're both very cute. So as you can see, the MinION sequencer is super small, which is super beneficial because it's very easily transportable. You can take it wherever. And even though it's this small, it can still give us our ultra long read lengths, and it's still able to give us back immediate data for rapid results. So this MinION uses Nanopore technology, and right here, this is a picture of what a nanopore is. It's basically a nanoscale hole in the membrane. So how this works, the MinION passes an ionic current through this nanopore, and then that current measures the change, the nanopore measures the changes in the current as molecules flow through it, and the device uses these changes in currents to be able to spit out the information and identify the molecule. So what did we find? So for our first hypothesis, which was that the pasture-raised chickens would have a more diverse microbiome, we looked at the alpha diversity, which is the diversity within a sample, and we used 3 different metrics. Here, this is just the number of species that were in each, which range from 35 to 240 between all the chickens. And then second, this gave us our estimated number of species because you can't count all the fish in the sea. And then this is the Earth-Shannon entropy, which gave us our community diversity using the abundancy of the species. Which showed us the abundancy of the species. So within the species level, we didn't find any significant difference between the 2 types of chickens. So we decided we wanted to go ahead and look at the phylum level. And here's just a quick refresher slide or to familiarize anybody with the taxonomic classification. So it starts at domain, it goes down to species. So we just looked at the species level, and then we're going to be looking at the phylum level. And I just gave an example of the human species. So we looked at the phylum level diversity, and as we can see over here, this is the pasture-raised chicken, the peas. You can see right away that there overall looks like there's more diversity on the pasture-raised side compared to the caged chicken side. And that actually is what we found. There were significantly more phyla found in the pasture-raised chickens. And you can also see from this slide that it's overwhelmingly purple and blue, which makes up the proteobacteria and the firmicutes phyla. And actually the ratio from the proteobacteria to the firmicutes for the pasture-raised chickens was about 7 to 1, while the caged chicken was about 1 to 1. And so since these 2 phyla overwhelmingly took up most of this chart, we decided to take them out to get a better look at the diversity. So this is what it looks like without those 2 out, and then again you can see there's a lot more variety and diversity on the pasture-raised side. And there are actually significantly more environmental bacteria present in the pasture-raised chicken. Another key thing that we found was that in the formerly caged chicken, they actually had 2 phyla that were not present in the pasture-raised chicken, which is the Tenerichides and the Fusobacteria. And Tenerichides is a phylum that is known to house some of the known chicken pathogen species. So because we knew this, we wanted to look into the chickens and see if they had any common avian pathogens in their system. So here's a chart. So we looked at these 3 main opportunistic pathogens that were in the respiratory tract. And as you can see, the pasture-raised had none, and then the formerly caged ones did have presence of the pathogens. And besides the pathogens, we also want to look at the most prevalent species that were found in each chicken. So here we have a list of the 12 most prevalent species found in both groups, and as you can see, these top 4, they actually overlap in both groups, which was an interesting find. And this first overlap is Bacterium avium, and you can see there's more abundance in the formerly caged, but for the Bacterium volunteum, there's actually more abundance in the pasture-raised chicken. So on to our second hypothesis, which was that each group would have more similarity within their own group rather than in between the 2 groups. So we looked at the beta diversity, which tests the diversity between samples, and we used the Bray-Curtis dissimilarity, which measured the dissimilarity between the 2 of them based on the number and abundance of their taxa, and it gave us these clusters. Up here, this is a clustering of the pasture-raised chicken, and then over here you can see a clustering of the rehomed or the formerly caged chicken. So because these clustered together, that means these are more similar to each other, and then this group is more similar to each other, and they did have some overlap. But as you can see, there's still a distinct difference. So for testing our third hypothesis, which was that the caged chicken would have high resistance to commonly used antibiotics, we use minimum inhibitory concentration, or also known as MIC, which shows us the lowest concentration of an antibiotic which has bacterial growth on it. So we did ours, we did 0 to 1,000, so this 0 has 0 antibiotics on it. You can see there's a lot of bacterial growth. As the concentrations get higher and higher, up to 1,000, you can see there's less and less growth present on the plates. So we found that the pasture-raised chickens were more resistant to polypeptide class antibiotics and beta-lactams, while the formerly caged chickens were more resistant to aminoglycosides, macrolides, tetracyclines, and sulfonamides. And over here, I boxed in the green. These are 4 of the most common preventative antibiotics that commercial facilities use on their chickens. And as you can see, there was some high resistance and resistance against these antibiotics from the formerly caged chickens who grew up in the commercial facilities, especially in the oxacillin and oxytetracycline here. There was also a lot of high resistance in the pasture-raised. Yes, in pasture-raised, okay. Now, so the overall takeaway from this, so some of the main key points that you should think of is that the overall diversity was not different between the pasture-raised and the caged chickens, but the phylum level diversity was significantly higher in the pasture-raised chickens. Also, the caged chickens had more Tenerifides and Fusobacteria prevalent in their microbiome, which was important because, like I said, the Tenerifides houses some of the known avian pathogens for chicken. And then the caged chicken did show higher resistance against some commonly used preventive antibiotics, which was an interesting thing to see. And then also in the future, we would love to add to our project by being able to test currently caged chickens, and then try and find some flow-raised chickens so that we can compare this to our data that we got during the summer. Another important thing to note is that any people raising chickens in here, I don't know, but it's important that you should try and let these chickens be more exposed to the environment, because this has been shown to give them a more diverse microbiome, which could give them higher resistance to infection. Okay. I'd like to thank Morningside College Summer Undergraduate Research Program for funding my research and allowing me to do this summer. I'd also like to thank Dr. Andy Moore for being such a great mentor and helping me with this project. I'd like to thank Stephanie Beaver and Mark Rogers, who they let us sample their chickens. I'd also like to thank any other people who helped us with the chicken sampling. And also a thank you to Randy Campbell for helping us set up this computer, which allowed us to run our DNA sequencer and get all of our data. And lastly, a thank you to all the chickens who let us sample and harass them. Okay. Are there any questions?
So was there any particular, type or breed of chicken you were working with, or just a random sample?
For the free range ones, there were multiple different kinds of them. Andy might have to help me with this. I remember there was the golden.
Golden Wyandotte. So the pasture-raised ones are all mixed breeds of all kinds of sorts, from Golden Wyandottes to Americanas, which Andy's holding there, to various common and other actually mixed breeds. The caged chickens, they were all white leghorns. Because they're medium-sized chickens and lay just a ton of eggs.
Sorry, I'm not very familiar with
Would
kinds.
would the breed play any type of role in, you know, are the microbiomes, you know, basically the same from breed to breed, or are there notable differences?
That's a good question. Since the free range or the pasture raised ones were all bunched together, there was just as much diversity between those as there was between all the white leghorns that were sticking together. So I don't think the breed really matters unless they have some really weird nasal cavity construction.
Because
It sounds like free range is better for the chicken. Is it worth $5 for me to buy their eggs?
I sell them for $3.
Well, so like you said, like the microbiome diversity is better for the chickens, which is seen more in the free range. So that would be less. So if they have a more diverse microbiome, it's less likely for them to like be sick or have an effect in their egg production. So some of like the chickens that we tested that were formerly caged that had presence of those pathogens, that possibly could affect like their egg production. Like I read some studies where like the chickens having pathogens affected like their eggs having like hairline fractures or like thin shells and stuff. So it is possible that the free range ones or the pasture raised would have like better eggs. It's feasible.
Do you think the changes you found are more from environmental factors or from genetic factors?
I think we think that it's more due to the environment that they were allowed to be in, because the formerly caged ones, and besides the 4 weeks that they were allowed to be in like a small penned-in area in a backyard, they were just in that slide, I guess they were born and raised and kept in these cages like all the rest of their life here. So they wouldn't really get any exposure compared to the pasture raised who are allowed to roam and like be in the grass, get the insects, be in the soil, trees, like whatever's on the pasture.
This is true for toddlers too, right? Let them play in the soil? Yes? Okay.
Any other questions for?
Speaking of toddlers, do you think that this might be, this is a question from one of my students, do you think that this might also be true for other domestic animals? Is this the, in other words, the freer they are, the more diverse their microbiome might be? Could you hypothesize that for us on the spot?
I would say yes, I think that's feasible, because I did in this slide where it showed like the comparisons between like humans, I also read some studies, like there were ones between like ducks, and I think I read one about sheep, and it has been like a trend that you see that the more diverse that their microbiomes are due to like their environmental exposure, like the healthier and better they can be. Any more?
Were you scared of the chickens, or?
Well, actually, this was my first time really like working with chickens. Like, obviously, I hadn't seen them before, but I did learn how to capture them. At least the pasture-raised ones, you kind of run up and like squish their wings out so they can't fly away. And like the more time we spent with them, I was less scared to like go and pick them up or hold them by their feet. So that was kind of cool. It's not scared of chickens.
Let's thank our speaker. Our fifth and final project is a project that was with Dr. Shannon Claxton, Caitlin McGovica, and Krista Zenk on attachment and sociosexuality, exploring differences based on religion and parental disorders.
So unfortunately, Caitlin and Krista were not able to make it today to show their project, but this also means I get just a second to brag about them since they're not here. And I just wanted to say that they really exceeded my expectations in this project. When we They are both interested in going to graduate school, so they said they wanted to do a project where they could write a paper and try to submit it for publication. And I thought, well, maybe, right? Like that 2 months is a pretty short period of time to get that done. And they really accomplished a lot during less than 2 months. They reviewed the literature, they learned a new statistical software, they ran so many analyses, which you'll see a little bit of that here. They wrote a 35-page paper, and last Friday we submitted it for publication. And so I'm just really impressed with that. So without further ado, here is their name.
This is our final presentation for our research project this summer. Caitlin and I couldn't be there for the final presentation, so we're just going to make this video going over everything that we worked on and our final results. So this summer we examined how sociosexual orientation and attachment were related, and specifically talking about sociosexual orientation in plain terms, it's one's or an individual's behaviors, attitudes, and desires about casual sex, so things like one-night stands, friends with benefit relationships, and people can fall onto like a spectrum in like to restricted or unrestricted. So having a more restricted sociosexual orientation means that they really don't participate in these casual sex relationships, and they prefer more of a committed relationship compared to somebody who's more unrestricted on their sociosexual orientation, meaning that they do participate in friends with benefits, casual sex, things like that. And in talking about attachment, you can either be insecure or secure in your romantic attachment, and this theory actually originated from a psychologist named John Bowlby in 1969, and he did an experiment talking about how parent and child attachment had a bunch of different implications for how people ended up later in life and like their experiences. And people have expanded on this idea, other psychologists, to find that this can go throughout the lifespan into romantic attachment. And so if you are insecurely attached, there are two different categories, so anxious or avoidant, which are the two insecure, and then securely attached. So again, we wanted to examine how attachment and sociosexual orientation are related and the association that they have together. So breaking down these two types of attachment again, so these are both insecure, and so on the left we have the anxious attachment, and these people desire emotional intimacy and they fear losing their partners. They can fall into these casual sexual relationships because they want that emotional intimacy, but they really do crave committed relationships. On the other hand, avoidant attachment, these people have a lot more of a selfish approach to relationships, and they prefer relationships where there's less emotional involvement, which is why casual sex is desirable to these types of people who are avoidantly attached. So our study specifically wanted to look at how parental divorce and religiosity might moderate the relationship between sociosexual orientation and attachment. And when we say moderation, we mean that how parental divorce can alter or change how sociosexual orientation and attachment function in their relationship. Moving on to our hypotheses for our experiment. So our first two hypotheses deal with attachment SOI, and we predicted that people who have higher attachment anxiety would also score higher on the SOI scale. And similarly, the same thing for avoidant attachment. So people who have higher avoidant attachment would also score higher on the SOI scale. For the third and fourth hypotheses, this was about divorce and attachment. So we predicted that people who have experienced parental divorce would also have higher levels of anxiety and avoidance attachment styles. For the fifth hypothesis, we predicted that people who have experienced parental divorce would have higher SOI scores.
compared to people who have not experienced parental divorce. And then for our 6th and 7th hypothesis, we expected that parental divorce attachment SOI, such that those who have experienced parental divorce would have a stronger association between attachment anxiety and SOI, and a stronger association between attachment avoidance and SOI than those who have not experienced parental divorce.
For hypotheses 8 and 9, based on previous evidence provided, we hypothesized that individuals with higher levels of religiosity will report lower attachment anxiety and attachment avoidance. Specifically, individuals who have low religiosity will show more anxiety or avoidant attachment behaviors, while individuals who have high religiosity will show limited anxiety or avoidant attachment behaviors. In addition, for hypothesis 10, we expected that individuals who have higher religiosity scores will have lower SOI scores than those who report lower religiosity. Specifically, individuals with high religiousness will show limited willingness to engage in casual sexual relationships, while individuals with low religiousness will tend to engage in more of those casual sexual relationships. Also, based on previous evidence, hypotheses 11 and 12 were that we expected that religion would moderate the association between attachment and SOI, such that those who experience lower levels of religiosity will indicate a stronger relationship between attachment anxiety and avoidance and SOI. We thought that the level of religiousness of an individual would impact the strength of the relationship between attachment and SOI. In our sample, we had 1,023 participants with an average age of 22.49. Of the participants, 68.7% of them were females. Our sample came from students at a large Midwestern university, as well as from an online survey platform called Amazon's Mechanical Turk. To assess sociosexuality, we used the 9-item Sociosexual Orientation Inventory Revised Scale. This measurement used 3 areas of behavior, attitudes, and desires to create the scale. Some example statements from each section are written on the screen. For our study, we created an overall scale of SOI, where higher scores indicated more unrestricted behaviors. To assess romantic attachment, we used the Experiences in Close Relationships Revised. This is a self-report questionnaire that measures 2 dimensions of attachment: attachment anxiety and attachment avoidance. Some example statements are given on the screen. For this measure, in our study, higher scores indicated higher levels of anxious or avoidant attachment behaviors. To measure parental divorce, we used one multiple-choice question that simply asked the marital status of the participants' biological parents. In our study, we omitted participants who answered never married and widowed from our sample. Also, we combined engaged with married, as well as combined divorce and separated. In order to assess religiosity, participants were asked to fill out a 2-item measure. The measure included questions to assess an individual's frequency of church attendance, also known as religious behavior, and his or her subjective religiosity. During our study, we combined these two measures into a single religiosity score, with higher scores meaning more religiosity.
Now moving on to what our results actually were. For our first two hypotheses about attachment and SOI, we did predict that there would be a higher or a positive relationship between anxiety and avoidance and SOI, and we did find that both of these hypotheses were supported, so that people with higher anxiety and avoidant attachment styles would also have higher SOI scores. For our third and fourth hypotheses about divorce and attachment, for divorce and anxious attachment styles, this hypothesis was not supported. So people who have experienced parental divorce did not have a higher anxious attachment style. And then for our fourth about divorce and avoidance, this one was marginally supported with a p-value of 0.056. So this hypothesis was somewhat significant. In our fifth hypothesis, we predicted that people who have experienced parental divorce would also have a higher SOI score, and this was not supported. There was not a difference. We also predicted that parental divorce would moderate the association between attachment and SOI. We broke this down into anxiety and SOI and avoidance and SOI. For the relationship between anxiety and SOI, there was actually a marginally statistically significant moderation effect with a p-value of 0.073. However, this moderation effect was the opposite of what we actually predicted. Specifically, the relationship between attachment, anxiety, and SOI were stronger for individuals whose parents were not divorced and for individuals whose parents were divorced, as you can see in the graph. There was no evidence to suggest that divorce moderated the association between avoidance and SOI.
Results for our religiosity variable. Our hypotheses for the relationship between religiosity and attachment were not supported, meaning that the level of an individual's religiosity was not associated with anxious or avoidant attachment styles. Specifically, individuals with lower religiosity did not show more anxious or avoidant attachment behaviors. As for our hypothesis regarding the relationship between religion and sociosexuality, this was supported. This meant that individuals who reported higher religiosity scores had lower SOI scores than those who reported lower religiosity scores. Specifically, individuals with high religiousness tend to engage in limited casual sexual relationships, while individuals with low religiousness will engage in more of those types of relationships. Although we expected religion to moderate the association between attachment and sociosexual orientation, our expectations were not supported. Our results showed that the level of religiousness of an individual does not impact the strength of the relationship between attachment and SOI. Our results of the relationship between attachment and SOI were confirmed by previous research. Specifically, anxiously attached individuals may engage in more casual sexual relationships in order to obtain intimacy with their partner, while avoidantly attached individuals may fear intimacy, leading to more casual sexual relationships and sexual permissiveness.
Our study found that divorce was moderately significant in predicting avoidant attachment, but we did not find a relationship between SOI and parental divorce. This means that people whose parents are divorced they actually did not have a higher SOI score compared to those whose parents are not divorced. This may be explained because sometimes after watching their parents go through a breakup, the person actually decides to become more restricted in their relationships because they don't experience the same thing. Another explanation of this is because quality of life actually improved for the individual after a divorce if they were living in a high conflict home, so a divorce could bring out a better life experience for somebody, which can then affect attachment.
For the relationship between religiosity and sociosexual orientation, our results showed that there was a negative relationship between the religiosity and sociosexuality of an individual, which was confirmed by previous research. This means that more religious individuals tend to have more restricted sociosexuality than those who are less religious. Limitations. The first limitation of our study involves the parental divorce variable. In our study, we measured parental divorce through the marital status of the participants' biological parents, which does not take into account if the participants are adopted or unaware of their biological parents' marital status. The second limitation involves the religiosity variable. Religiosity tends to be more influential during the adolescent years, which means age is a limitation of our study because the participants of this study were within their emerging adulthood or adults, aged 18 to 29. The third limitation of our study is the cross-sectional design due to the data being collected at one time and not longitudinally over time, which makes it unclear whether attachment or SOI comes first in the relationship.
Some future directions for research to expand on these topics and to better improve this area. So first we suggest for an idea would be to have a longitudinal research study. Because our study was cross-sectional, it's hard to make any causal claims because we did just take a snapshot in time of people's behavior and attitudes and their romantic attachment. But if you follow people over the course of 10 years, you could really understand better how sociosexual orientation and attachment are related and which causes what, and that could just really improve this type of research. The second one has to do with parental divorce, so when parents get divorced, because we noted earlier in researching studies that they say if a parental divorce happens before age 10, that has very minimal effects compared to if a parental divorce happens after age 10. So having more information about how old a child was when their parents got divorced would be really useful. The third is a religious denomination question, asking what denomination somebody is, so then we could figure out more to what extent do each denominations have on, what effects do they have on the SOI for each person.
There are several ways in which our results connect with society in this field of study. The established connection between romantic attachment and sociosexual orientation helps us to understand how an individual's attachment within romantic relationships may be associated with the individual's sexual experiences. Additionally, this study confirms research suggesting that parental divorce may not have a long-lasting association with relationship attachment. Furthermore, this study shows that higher levels of religiosity are associated with a more restricted sociosexual orientation, suggesting that religiosity may help predict engagement in casual sexual relationships and experiences. We hope you enjoyed listening to our summer research project. Thank you.
The one thing that stuck to me the most was just speaking with like the interviewees. I mean, they had really interesting stories about like how they came here or how they built their lives here, and it was just very, very impactful. Just because I mean our community is very diverse and they come from all over. I love my professors. I mean, my professors have helped me encourage me to do the best that I can. They helped me like just develop my like just abilities like in general, like my writing, my just like overall learning experience, and I really appreciate everything that they've done. And I'm also like just like everyone here, like everyone feels like a family. So
I'm a pre-vet student, so I just thought it'd be an awesome opportunity to be able to work with animals this summer, and it turned out being a really awesome and fun project for both of us, so it ended up really great. One of the biggest things that I learned doing this research would probably, I learned a lot about chickens in general and about how microbiomes, how if somebody has a more diverse microbiome, which in the cases of the chickens and in human studies that we looked at, they have more protection from infection, which was really interesting to me, so I really liked learning that aspect of it. So yeah, it was actually super interesting. So I knew what chickens were obviously, and I had seen them before, but I never actually physically worked with them. So when we took our samples from the inside of the chicken's mouth, I was just super surprised because on the upper part of their mouth they have a giant slit in the middle that leads straight to their nasal cavity, which was super interesting. I honestly didn't even know any type of animal had that type of thing inside their mouth, which was really cool. Also, I learned how to handle the chicken more. I was really timid at first trying to handle them because I didn't want to hurt them. But then Dr. Moore showed me how to you kind of crouch in there, put your hand on their wings so they don't fly away, and then you just kind of grab them and you kind of hold them honestly like you would a puppy. So it was just really fun. Having a smaller school, I just have better relationships with my faculty. It's shown with me and Dr. Moore, I was able to be comfortable around her and have her teach me new things and be open to learning things, and I just have a better relationship with her overall too. I would say that I'm really happy that I did this experience because I learned so much, and there's so many different things you can do with this summer research program. You can really find something that pertains to your interests and pertains to something that you might want to learn about, so you can make it fun and make it your own, and it's just been a really good experience.